an intern like that.
They have one, they have one. Yeah, they just, the new one came on.
Good morning, volunteers. Bruce, I'm afraid that you're meeting with the opening of the impeachment hearing. Did you realize that? Actually, hearings going on. Anyway, welcome on this snowy day. And it's a lot of our usual introducers are not here, so I got Bell's introduction gig. I can add a little something to the introduction to Bruce that I think is sort of interesting about him and about the college. Is, well, it's supposed to be. Can you hear?
Yeah.
I'll kiss her. And so, as I said, I was going to add a little bit to the usual introduction for Bruce. In the late 1970s, Rush Medical School began a first-year medical education program at two liberal arts colleges, Knox and Grinnell. The idea was this would humanize future doctors. And so the college then, through Rush, hired medical school professors. That's how Bruce got to Grinnell. Started in that program, and then when that program ended, he stayed on in our biology department, luckily for us. So you were here last week, and you heard the beginning of his lecture series on the virologist's point of view on influenza. And it's a great pleasure to welcome Bruce back and with the continuation of his lecture. Thank you, Bruce.
Thank you.
Well, thank you all for coming. I just figured half the people who were here last week had given up. So I'd like to know there's a reason. Although, missing this for politics, I can't believe that. Anyway, last time I introduced you to viruses in general, and then began talking about the influenza A virus in particular. And I realized that there's one thing I forgot to mention right at the beginning, and that is there are actually 3 influenza viruses in humans, cleverly named A, B, and C, but you know. A is the one that is the seasonal flu and also the pandemic flu. And this morning I'll be telling you about how influenza A virus changes to produce both of those types of flus. Influenza B also causes a typical sort of influenza in humans, but it doesn't have the genetic variability that produces strikingly different kinds and pandemics and so forth. And then C is essentially not noticeable if you get infected with it. So that's why I just am focusing on A. Let's see now. I should remember how to do this. Okay. Last time I introduced you to the influenza virus and its structure and paid special attention to the spikes on the surface membrane of the virion, the hemagglutination spike that is the attachment device to get the virus attached to the cell surface, and the neuraminidase spike, which is operates at the other end of the life cycle to facilitate the virions leaving the cell. And so I want to start this morning by talking about the influenza, quote, life unquote cycle, that sort of allows me to ignore the question of whether viruses are alive or not. Quotes are a wonderful thing. Okay. So the typical viral reproductive cycle involves getting the virus in, getting the genetic material free so that it can be expressed, and then copying, and then assembling or building from scratch the new viruses that will then be released. So the first step is called, surprisingly enough, attachment. And this is the most important thing in the virus life cycle, because the virus has to attach to the right cell. And what we speak of in these terms is viruses have host ranges, and the host range can be extremely broad, like rabies virus, infect the nervous system of virtually every mammal. So it has a very wide host range. Influenza has a moderate host range. It can attach to the respiratory tract of birds, it can attach to respiratory tract of pigs and humans, and some types of it can attach to bat cells and so forth. And then there are other viruses that have exquisite host range demands. There is one that infects mice, but it only affects newborn mice, and it only infects newborn mice that are a couple of weeks old. Okay, so it's got a very narrow host range, but part of what makes influenza to me so interesting is its host range is broad enough to cause all kinds of trouble. And I like trouble when it's viruses that are not in me. Okay, so as I said last time, attachment, let's see, attachment to the host cell surface is the first step, and then penetration, and that is the entry of the at least the genetic material, possibly the whole virion, into the host cell. And that's followed by uncoating. And uncoating is release of the viral genome. The viral genome is within a capsid, and the capsid may be spherical, so it's a ball with the DNA or RNA inside of it, or like influenza, as you'll see, it's more like the very first virus I showed you a picture of, which was tobacco mosaic, and it was a rod-shaped virus with proteins wrapped all around its outside. And influenza has a similar sort of organization. But the uncoating is necessary for the genome to be expressed. It's getting rid of those capsid and nucleocapsid proteins. Then the next stage, or next two stages, are expression of the virus genome. And the first thing the virus has to do is make new proteins. And these are proteins that will then make new copies of the virus genome, as well as the proteins of the capsid and proteins that will be embedded in the surface membrane of the cells if it's going to be an enveloped virus. And I am going to mercifully sort of skip over the details of those steps, because I didn't think you really wanted a whole semester's course in molecular biology before I went on. Then, now see, I keep, they need bigger buttons, or I need to be less clumsy. After this synthesis of all the necessary proteins and new copies of the virus genome made, then new viruses are assembled from those constituent parts. And that doesn't require anything from the host cell except providing the materials with which to build the new virions. And then maturation is a step that occurs in envelope viruses. The assembly of new virions in envelope viruses creates what we call a nucleocapsid, which is nucleic acids and capsid proteins. But the membrane that's going to form the envelope of the virus has to be modified by introducing viral proteins, like the spike proteins that I've already mentioned, as well as proteins that are called matrix proteins. And they bind to the inner surface of the envelope area of the membrane, and they sort of provide the location where the virus nucleocapsid will come up and then leave the host cell and derive their membrane envelope at the same time. And so the last step is release of the virions. That can be, in the case of naked viruses, simply blowing the cell apart. In the case of envelope viruses, the nucleocapsid gets surrounded by a membrane, often a cell surface membrane, but it can also be internal membranes in some viruses. And so cells like that can sort of release virus for a period of time until they damage so much of the host cell's membrane that it then disintegrates. Okay? And I forgot to mention again, if you have questions, just raise your hands and we'll the royal we, I will attempt to address them. But there's no point in your sitting there becoming more and more befuddled when you might have asked a question that would clarify things, I hope. Now talking specifically about influenza virus, the host range for influenza virus is established by the binding of the hemagglutination spikes to an N-acetyl neurominic acid that is in the proper linkage or connection to the next sugar in a chain. Sugars are essentially chains of carbons, usually 4, 5, 6, 7, 8 carbons, and they often fold back on themselves and form a ring with one of the last carbons sticking out. And because there are so many sugars, there are so many carbons in each sugar, there's a whole variety of ways that they can attach together. And in this case, the terminal sugar has to be attached in the proper fashion to the next sugar in order for the hemagglutination spike to bind to it. And I showed you last time that the spike is actually a trimer and with a ball of protein at the end of each of the three components. And that region is what fits, when we say it like a lock and key. It has to fit properly on these terminal sugars. I want to emphasize that the cells that have this configuration are in the respiratory tract. They line the respiratory tract from the throat down into the lungs. And so influenza is a respiratory disease. I think I said this last time, but it's one of my pet peeves. When people talk about a stomach flu, no such animal. You can have a virus in the stomach, in your gastrointestinal tract, but it ain't influenza A. The binding of the hemagglutination spike, shown here in cartoon. Here's the cell surface membrane, the new host cell, and here's the viral envelope membrane, and the spike is sticking out so that these more or less spherical components are going to bind to the host cell membrane. And when that happens, it triggers a very large conformational change in the spike protein. This red region here has a hinge, and it folds back on itself, and it also allows what's called a fusion peptide, shown here in yellow, to pop out. And the fusion protein will embed itself in the host cell surface as the head of the hemagglutinin folds back. And so, as you can see, as this folds back, it's going to pull the two membranes together, pull the virus down to the cell surface. And when that takes place, this fusion peptide gets embedded in the host cell membrane and magically causes the host cell membrane and virus envelope membrane to merge into a single membrane. Well, I won't go into it, but it's a very puzzling reaction because membranes are designed to keep things out, generally speaking. And so getting two membranes to fuse together is a neat trick. Okay, then penetration is the next step, and the binding of the host cell surface and the virus envelope triggers a process we call endocytosis. And endo means within, cyto literally means a hollow vessel, but it means cell. And then osis is a state or condition. So you're going to learn all kinds of I always think that people who are going to go into science, specifically biology, should have Latin and Greek in high school. Make it a lot easier. Endocytosis is bringing the two entities, the host cell and the virion, together and introducing the virion, at least the nucleocapsid, into the host cell. And what happens in it, for crying out loud, what happens in endocytosis is that the membrane that's now fused with the virus and the host cell comes around and folds inward so that eventually it's a sac that then pinches off on the inside of the cell. And so the virus has entered by causing the cell to engulf it. Okay, that's endocytosis. That produces what's called an endosome, endo within and some from soma meaning body, and it's a sac containing now the virus nucleocapsid. And the endosome is carried to the nuclear membrane where uncoating occurs. And that's one of the neatest cellular reactions. There are actually motor proteins and long fibrous proteins, and the motor proteins attach to the endosome, and then they literally walk down the road, if you will, to the nucleus. And then there are others that walk back the other direction. So, you know, there's none of this floating around stuff. Anyway, the endosome gets carried to the nuclear membrane where it fuses with another, excuse me, another vesicle called the lysosome. And lysosomes are for digesting material in the cell. And so when they fuse, causes the pH of the combined vesicle to drop, and that causes the membrane to break down and releases the 8 pieces of single-stranded RNA and their associated proteins, releases them into the nucleus. And that's important because the nucleus is going to be the site where protein synthesis machinery is built, messenger RNAs, and where new virus genomes are going to be made. And so here's this process of attachment, formation of the endosome, fusion with the lysosome, and release of the 8 pieces of nucleic acid. Yes?
I'm just wondering how you observe that process through the microscope, or how do you know that that's the process?
Okay, good question. See me afterward and I'll try to know. Excuse me. You can see the formation of the endosome and the fusion with the lysosome in a light microscope. You can't see what has caused this process because the virus itself is too small to see in the light microscope. But you can see it in the electron microscope. Likewise, the assembly process you can see in the electron microscope the details of the assembly and release, but in the light microscope you can just see the formation of this bud, as it's called, and then the bud being lifted off. Okay, so, and then if you want to go the molecular route, you can show that there's the envelope and the cell surface membrane have attached to one another and then fused and so forth, while you can isolate these various components. Does that kind of get to it?
Yeah, sorry.
You do have to take a lot of faith when you're working with things you can't see. And so that means virologists are extremely clever because they can figure out what's going on. They're not as clever as organic chemists, though. They're the people I live in awe of. And that's aimed at Professor Wubbles there. I regard organic chemists as even smarter than physicists. That's saying, my son's a physicist, so, and he told me that he didn't take any biology in college because it was too easy. Which, you know, okay. This cartoon shows you what the virion, back up there, what the virion looks like. It's got 8 pieces of RNA, and each piece of RNA has a structure like this. The RNA is surrounded by capsid proteins, and then at the end, where there would be the ends of the piece of RNA, there are 3 proteins that are attached called PB1, PB2, and PA, standing for polymerase. And polymerases are enzymes that hook subunits together. Poly is many, and mer is a unit, and ase means it's an enzyme. So it's an enzyme that attaches subunits together to make a larger molecule. And 2 of those, in the case of influenza virus, operate at a basic pH, and one of them operates at an acidic pH. And that difference in pH requirement allows for staging of synthesis of new RNAs, so that the messenger RNAs that will be used in protein synthesis are made first, and then nuclear, new genomes are made second. So the basic, the basic proteins are involved in this first step. This is the so-called central dogma of molecular biology. The term central dogma was coined by Francis Crick of Watson and Crick, or as we call him, Crick and Creep, because Watson is, to say he's not politically correct is to give him a lot more credit than he deserves. Anyway, and Craig has one of the most original minds in all of biology. He's the one who could see how things had to be in order to get to where he had to get. And then he wasn't much of an experimentalist, so other people would then prove that he was correct. Okay? And the central dogma is that the name quick applied to the processes of copying a genome to make new genome and then expressing the genes within the genomic molecule. Dogma, as you know, is something you have to accept on faith, and so it is absolutely contrary to what science is. And so the central dogma is typical British witticism, and Crick does it a lot. Anyway, the central dogma says that information is stored in the form of a genomic molecule. And the genomic molecule does not have these big springs attaching it. It's all a continuous strain of DNA bases. The first step of the central dogma is not shown here, which is copying this DNA molecule to make new molecules. That's called replication. Expression of the genome is first by transcribing the information here in the DNA molecule, or in the case of influenza virus, the genomic molecule, into an RNA molecule, which is complementary to the genomic molecule. Complementarity means that only specific base pairs can be made. That's the magic of DNA. And it's one of the reasons my students were extremely fortunate to have decided to come to Grinnell College, because the base pairs are adenine and thymine, which spells at, and the other base pairs are guanine and cytosine, so they are at Grinnell College. So they'll never forget. Not that they'll forget anyway, but I discovered that. That was very clever. Anyway, messenger RNA is the complementary information to the what's called the template, either RNA or DNA. The next step is translation, where the information contained in the sequence of bases in the messenger RNA is used to determine the sequence of amino acids in a protein. Okay? And the thing I want to point out here is that the messenger RNA is defined as plus strand. Okay? Its complement is therefore minus strand. And this is important in virology because the genomic molecule can be plus strand DNA or minus strand DNA, or both if it's double-stranded. The RNA of the genome can be plus strand or minus strand, or both. There are double-stranded RNA viruses. And so by defining the strand that's used in the process of translation, we can establish the plus strand. Its complement is therefore minus strand. And I'm going to skip over this sort of stuff. I don't think for this talk, given how long I've talked already, I'm going to go into how the RNA of the virus is. Now, you're going to make me go through all that?
This is a real genuine question.
Okay.
Those two strands, are those the two strands that form the double helix that we hear about?
In DNA, yes. And RNA can form a double helix also. But RNA is, because of the chemistry of the sugar in the RNA strand, it's very easy to break. And so it's not like the DNA double helix. It's a much more fragile molecule. Okay, now we get to the stuff that is possibly of interest to you, unlike all of the preceding. How do new strains of influenza virus arise? It should be clear that the 1918-19 pandemic was caused because a new virus appeared. And the question then is, where did it come from? Okay. A similar question of a much less tragic nature is, why does the seasonal virus seem to change every time it comes through? Which is the reason you have to get a flu shot every year. So I'll start with that one because I think it's the one that has most influence on our lives. Pandemics come around every 15 or 20 years, but the seasonal flu is year after year. And the process that causes new virus strains is called genetic drift. And as you know, drifting is a sort of gentle movement away from a point. You know, if you forgot to tie up your boat on the lake, it may have drifted away by the time you come out in the morning. And it probably didn't turn itself around and turn the motor on or raise the sail and move off. It just floated away. Well, genetic drift is a similar kind of process where there is a small change in the genomic molecule of the virus, and that change occurs year after year. Are you familiar with the game that, at least when I was growing up, was called telephone?
Yes.
Okay, where you give a word to the first person of a line, and they whisper it to the next, who whispers it to the next, and so forth. And the fun thing about the game is, at the end, the last person will have a word that may bear no resemblance at all to the original word. And it wasn't a leap from the first word to the last word, but a series of small changes as the word is whispered along. Okay? So that's the kind of change that we're seeing in genetic drift. Incremental changes of a single amino acid at a time. And if that change of amino acid occurs in the knob at the end of the hemagglutination molecule, it may change the binding of that to a host cell. One in the neuraminidase may affect how it works, cleaving neurominic acid off of sugar chains. The error that produces a change in amino acid is a result of the process of making new RNA. If we think of the economy of the host cell will have a DNA molecule as its genome, and that DNA molecule must be copied absolutely faithfully so that each daughter cell after division has got a perfect copy. Because even one mistake may be lethal to a daughter cell. So the process of making new DNA involves several steps that proofread as new DNA is being synthesized. The DNA polymerase attaches a new base to the growing point and then checks it, and if it's wrong, it takes it off and puts another base on, tries again. That's proofreading. I'd like to say it's what our students were supposed to do with their papers, but of course they rely on the idiot computer to do it for them. So now instead of spelling errors, you get these incomprehensible sentences where the computer has inserted a totally different word than what the person writing the paper intended. Anyway, so I always have to explain what proofreading is because they never do it. Okay. RNA in the host cell is made in enormous abundance. Thousands of copies of messenger RNAs are made. If you make a mistake in one, there are lots of others. So they don't bother, RNA polymerases don't bother to check. They just plow on. Well, the genome of the influenza virus is an RNA. It's an RNA polymerase that makes a copy of it, and it makes mistakes once in a while, and those mistakes may then cause a change in amino acid. That's genetic drift. It's a result of the inability of RNA polymerases to check what they've done. Is this just telling me the time, or I'm supposed to stop?
It is the time, yeah, for our break.
Oh, already? All right. Hope you've worked up an appetite. Because things are going to go much faster when we get back.
So we have a 10-minute break.
Okay, can you hear me?
Yes.
Oh, okay, because the little red light did not turn green, so. Regarding the intercourse evaluations, that isn't good.
Better go sit down, maybe that'll go away.
All right, we'll try it again. Grinnell College required end-of-course evaluations, and that was fine, they should. But I have to tell you, the comment that pains me to this day is that one student said, "You're not as funny as you think you are." And And much earlier than that, actually, I had a student write on an end-of-course evaluation, you make me want to buy a gun. And this was far enough away that we didn't call in the police. And it's partly because I couldn't interpret that. Did you want to do away with yourself, or did you want to shoot me? So anyway, moving right along. Genetic drift that causes these small changes on a kind of yearly basis produce the seasonal flu, the flu that comes around every year. And H3N2 is the seasonal flu that we have had circling around since 1968. And so it's had lots of changes year to year. And these changes may cause epidemics. That is a localized outbreak with a lot of people getting sick. But they don't cause pandemics. They don't circle the world. And it's partly because the changes are so small. So how do new strains appear? I've talked about H1N1, which was the strain that caused the 1918 pandemic. 1968 was the Hong Kong flu. And I'm going to waste more of your time, that's why I'm never going to finish. In my textbook that I used to teach medical microbiology when I first came here, there was a diagram that showed the dispersal of the Hong Kong flu virus in the United States. And it probably came from Vietnam with a returning sailor, because the first incidences in the United States were in San Diego. Then there was a big arrow across two-thirds of the country to this place called Grinnell, Iowa, G-R-I-N-E-L. But I recognized it. Turns out there was a music festival here, and this carrier brought the virus to that music festival, shared it with all the other participants, and then there is this blossom of lines going all over the country. And so Grinnell was apparently the epicenter for the movement of the Hong Kong flu pandemic in the United States. Somebody stole that book, and I have searched for years, and I can't find that figure, even though I think it came from the Centers for Disease Control. Anyway, so how do we get from H1N1 to H3N2? We get there because, as I mentioned, influenza A virus can infect us, it can infect birds, it can infect pigs, and so forth. And new combinations of hemagglutinins and neuraminidase spikes come about because some cell is infected with two different influenza viruses at the same time. And specifically, a bird virus and then a human virus. And the process of getting out of the host cell involves the different genomic molecules of the virus to move up to an area of the cell surface membrane that has been prepared by having a layer of so-called matrix proteins laid down on the inside, and then H and N spikes on the outside of the cell surface membrane. Well, there's no mechanism that makes sure you get one of each, or that the 8 you get, in the case of a doubly infected cell, are from just the avian virus or just the human virus. It's a crapshoot. And sometimes you'll pick up, you'll make virions that have the avian H and a human N, and so forth. And that's how you get these recombinations. Recombination is a new combination of genomic components. And it isn't just the H and N that get shuffled around. Because these are such enormous changes, this is called a genetic shift as opposed to genetic drift. Okay? These new viruses may cause a pandemic because it's very possible that this new combination is one that has never been encountered by us, or the last time that combination was around, none of us were. It was before we were born. Okay? This says essentially the same thing. The other thing I want to point out is that this process of getting all the subunits, all the RNA units up to the membrane and budding them out is not controlled in any way. And so lots of what we call defective interfering particles are produced. They've got 6 RNA molecules instead of 8. They've got duplicates of one type and are missing another, and so forth. So they may be able to attach, or they may interfere with the attachment of other virions, but they don't make new virus because they're defective. And influenza makes lots of those, which is a saving grace for us. Okay. I think I did that. Okay. So this is how genetic shift, or it's called, if you're looking at it from a point of view of the immunologist, it's antigenic shift, same thing. We have a human virus and a bird virus, an avian virus, infect a pig, which is perfectly happy to host both of them. And that reshuffling occurs to make a new virus that's got some human virus and some pig virus components, and that new virus then infects humans. This is the reason why many pandemic viruses arise in Asia, because there's a lot more pig production per capita in Asia than there is, say, in the United States, and the pig raising tends to be on smaller non-factory farms, and these same farms often have a pond, and so wild ducks can come into the pond and poo a lot, and the pigs, when they drink the water, take in that material with its viruses in it, and the caretakers of the pigs give them the human virus, and the result is the shifted virus. And this shows you the same thing. This is a reaction that we greatly fear. It is the human H3N2 virus that is currently here, and a bird virus, H5N1. And they may combine with each other to make a new virus, which is H5N2. And that virus is not transmissible to humans. As we say, the reasons for that are beyond the scope of this talk. However, it is entirely possible that a sufficient change in this H5N2 virus will make it transmissible to people, and specifically from person to person. That would mean there would be one hell of a pandemic, because the times that this virus has gotten into people, its lethality is enormous. 90 to 100% of the people, human beings who have been infected with this, have died. So we really don't want it causing a pandemic. And that's there isn't really anything we can do to prevent this combination from being made, because it's being made all the time. We're just hoping that the other mutations in other components of the virus don't occur, so that it's transmissible from person to person. And so we're due for a pandemic, incidentally. They come around every 15, 20 years. And let's just pray it's not that. Okay, epidemics and pandemics. The seasonal flu is usually associated with a high fever, tiredness, which is not surprising if you have a high fever, headache, dry cough, sore throat, runny, stuffy nose, and muscle aches. Notice, there's nothing in here about vomiting, diarrhea, stomach flu, all right? It's a respiratory disease. This is a seasonal flu, and about 5 to 20% of the population get seasonal flu. It's primarily the young and the elderly, and it's because their immune systems are weaker. They're not fully developed in the case of infants, and in us old folks, they're giving up. So that's why we get the flu. And people tend to talk about it like it's no big thing. 200,000 people a year are hospitalized because of flu-related complications. Okay, that's a lot of people. And deaths from flu-related causes range from about 3,300 to 4,800 since the 48,000, yes. This is with the H3N2 virus. The average is 23,000 a year. So this is not inconsequential. Okay, it's nothing like the 1918 pandemic, but if it's your grandma or you, it's consequential. I've shown this before, but I want to show you again. The 1918 pandemic with the H1N1 virus a rib-cracking cough, high fever, dark spots on the cheeks, cyanosis because you're suffocating, because your lungs are filling up with a bloody, frothy substance, and there are massive pulmonary hemorrhages or bleeding. Okay, so it's quite different from the seasonal flu. And as I told you last time, the mortality and morbidity were much higher than we see in seasonal flu. I was asked over the break, how did it kill? And I don't think I showed you this before, but it is thought that it killed by what is called a cytokine storm. And cytokines are molecules produced in the immunological response. And a cytokine storm means a vast overproduction of these cytokines. And these cytokines then cause the immune system to go nuts, and or maybe it is already nuts, but they interfere with the immune reaction to the infected cells. Young adults have stronger immune systems than the elderly or children. And so here is an example of a good thing that turns out to be a very bad thing. Because young adults have very healthy immune systems, the cytokine storms caused by infection are much stronger and therefore cause the infected person to be much sicker and possibly to die. Young people and the elderly have weaker immune systems, therefore our cytokine storms are less stormy and fewer of us die. Okay? So that's why there was a different population in the pandemic situation compared to the later one. A gentleman came up over break, and I realized I didn't get his name, but he said that his parents, I guess, and their family were alive when the 1918 pandemic came around, and none of them got infected, and he was wondering how that was possible. Well, it's possible in part because the virus circled the Earth 3 times. And the first wave, it produced something akin to the seasonal flu. It was nothing like the second wave, which was the devastating pandemic. And then after that, there came a third cycle around the world, and the virus was far, far milder, and then it seemed to disappear. Okay? So, why it became far, far milder is not exactly clear. We think we have some ideas, but we haven't demonstrated it. And one of the sort of fraught questions is that there are people who would like to experiment with the H1N1 virus to see if they can alter the virus from its fairly benign form to this incredibly lethal form. And it certainly can be done with molecular genetics. It can be done in the lab. But there are many people who think that that's one of the experiments that ought not to be done. And unfortunately, there's always somebody, and I hate to be chauvinistic, but they tend to be Chinese who don't regard the safety issue of containing the virus quite as seriously as we do. Anyway, that's the way this progressed. In 1978, H1N1 appeared again as a seasonal virus, and it's still being passed around as a seasonal virus. And in fact, the virus vaccines that we're getting are aimed against both H3N2 and H1N1 and influenza B virus as well. H1N1 returned again in 2009, 2010, and it was called the swine flu virus, and I liked this little cartoon, swine flu and mad cow. Anyway, the virus began looking like a pandemic virus, and young adults rather than babies and golden agers were infected, pregnant women were infected. It was very virulent, and there was enormous effort made to produce vaccines against this virus so that if it was pandemic and was going to come around, we'd be able to get people vaccinated. It disappeared after a year. And again, they don't know why, and they have all of these vaccines that have nothing to attack. And there's a bunch of yiping about all the waste of producing hundreds of millions of vaccine doses, but there would have been a lot of griping if they hadn't produced them and they came back. So it's an example of where our government, was damned if it did, damned if it didn't. And I personally am much happier to spend money making the vaccines in a lot of other ways that they spend money. Okay, here's a breakdown of the various pandemics and the estimated deaths that they caused. This table omits a mild pandemic that came through in the 40s. You may recall we were busy with other things, and so didn't pay that much attention to it. And this shows you the persistence of these viruses. H1N1 from the pandemic, H2N2 caused the 1957 pandemic, and it only was around a couple of years. Then the 1968, the Hong Kong flu still is out there. And, excuse me, there are now 2 different H1N1 viruses circulating. And it was the 2009 one that looked serious, but was only around for 1 season. Okay. The bird flu. I already talked about this. This is where we fear that an avian virus and a human virus will recombine along with the fear that they will enter the human population with the ability to be transmitted. These viruses are bird viruses, and they are highly pathogenic in chickens. And I'm sure most of us recall that the control for this was to kill all the chickens. And so egg prices went way up, chicken parts went way up in price. It had a significant economic impact, but it did also have the impact of reining in the bird flu epidemic. Thus far, these two viruses are bird viruses. They're not adapted to humans, so they're not transmitted easily from person to person, but they do cause a really nasty, highly lethal infection when it does happen. Okay, prevention and treatment in 5 minutes. All right, flu shot, get one every year. This year it's against H3N2 and H1N1. Get one every year. Remember genetic drift. Get one every year. Is that subtle enough? You too.
How do they know what to, how do they make the vaccine for this year or whatever?
It's called an educated guess. And truly it is, because it takes about a year to make the number of doses that are required to immunize people in the United States, and then the vaccine is sent all over the world.
Right.
And so they look at what's been going around, what it looks like in Asia and so forth, but it's still an educated guess. And they usually get it kind of right, but often it happens that it's not a good fit, or it can be the entirely wrong virus. But they did the best they could. Yeah, Harriet?
What I was wondering about, obviously antibiotics don't work because they're not antiviral, but there are things coming out like Theraflu. How does that react? I mean, it's not a cure, but what does it do to mitigate some of the problems?
Okay, that's about 2 or 3 slides down, if you'll forgive my not answering directly. I do want to show you this, because even the best of flu vaccines will only reduce infection by about 50%. And it's often much lower, sometimes as low as 10%. They really missed in 2004-05, okay? And so there is, unfortunately, a trend of people saying, man, they don't work anyway, I'm not going to get a flu shot. Well, they work some. I would certainly take 50% as opposed to 0%. And they don't even hurt, so. Anyway, types of flu vaccines. There are what are called attenuated virus strains. These are viruses that have been manipulated in cell culture for a long time, many passages in cell culture, and that passaging weakens them. And so they get to a point where they're no longer able to cause disease, but they are still fully intact viruses that reproduce in your cells. And that means they're highly immunogenic. So that would be the best possible vaccine. Unfortunately, these can back mutate and make people sick, and so, they're useful, but cautiously. Inactivated virus vaccines are vaccines in which the virions are treated with chemicals. Glutaraldehyde is one, or physical means, heat and so forth, that alter the structure of some of the proteins in the virion so they can't reproduce. Now this one is safer, but it's not as immunogenic, and so that's the trade-off. And then subunit vaccines are composed of selected proteins that cause an immune response in the intact virion. But these proteins are made by molecular genetic techniques. They're produced by bacteria. And they don't work very well because the proteins are not seen in the context of the whole virion, okay? But they're safe. Now, there is a great effort to produce a universal influenza vaccine. And this would be a vaccine against all the seasonal flu types and all the animal strains that can also infect humans. And the problem is that they have to have an immune response to something that is the same in all these virions. And that same thing would be here in the stalk of the trimer. Here's the head, and then here's the stalk. And we generally don't make any response to this region. All the response is to the head. And so what they're working on is having the stalk with its proper configuration of the protein without having the heads. And they're taking a non-immunogenic head from another virus and attaching it to the stalk and so forth. There's a lot of work being done on this. And this would be wonderful. You know, you might only have to have 1 or 2 shots and you're good to go. Now, Harriet's question was about drugs. There are antiviral drugs to treat flu, and they target 2 different parts of the virus reproductive cycle. 3 of these drugs interfere with the activity of the neuraminidase spikes. Neuraminidase cleaves off neurominic acid when the virus is budding out, so it can't, so the virion can't go back and grab on the surface again. And these are what are called competitive inhibitors. And so it's a matter of having enough of the antiviral drug to interfere with all of these. Okay? And they currently shorten the duration of symptoms by about a day, which, you know, that's a day. But they're not a cure-all. All right, 1 more minute. There are antiviral drugs that target what are called the M2 proteins. And 4 M2 proteins come together to create a pore through the cell surface membrane. That pore is necessary to allow transport of hydrogen ions into the cell, and that gives energy to the cell. There are two currently available to block the pore. The chemical agent goes into the pore and gets stuck, so it can't transport hydrogen anymore. Well, viruses are not dumb, and so the seasonal viruses have developed resistance to both of these drugs, and so they're really of very limited use. And methinks, yes, that's it. Thank you.
Thank you very much, Bruce. And two things happen as a result of your being here. We give you this wonderful certificate of appreciation, which I know you will post immediately on your office wall. And also know the contribution to the Art Hyman Book Fund here at the library. And we do thank you. I guess we could get the flu, but at least we'll know why. Just a word, next week is the end of our fall semester bucket courses, and you're going to be subjected to me. Another one of these Americans we should know more about, single lectures, Henry Clay and James K. Polk, and they butted heads in the 1844 election, which Polk barely won, and the course of American history was significantly altered because of that. So that's what's on for next week. Thank you.